bring them together. How do they know which ones to bring together? They don't. It's random. It's completely random. One of them binds to one of them binds to another. And they bring them together. You can think about rag will only bind to, rag 1 will only bind to the 12 base first baser, rag 2 only to the 23. I don't know if that's true. But for our purposes, the rag 1 and the rag 2 heterodimer bind and bring these things together at these junctions. The other one I care about is this terminal deoxyribonucleic transferase, TDT. This enzyme basically just adds nucleotides onto a DNA strand. Doesn't care. So whatever it grabs hold of. If it's a T, it puts it on there. If it's an A, it puts it on there. But what I care that you know is that when we go in there and we bring these two DNA pieces together and we're trying to put them together, we're going to cut the DNA in both places. And that's an opportunity for this TDT to come in and start adding stuff. It just adds whatever it wants. It's not template driven, it just adds whatever it finds. So it'll add a T, it'll add a C, a G, whatever's close by. And when it does that, it adds junctional diversity before you actually get the V hooked to the J, the D to the J, the V to the DJ, if that makes sense. These others are involved in some other things, in ligating, etc., but we're not going to worry about it. This shows you, I'm just going through a little thing here, just so you can visualize what's going on. One rag complex binds to 23 base pair spacer, one binds to 12, and what we're going to see is the DNA is going to bend and bring these two together. They then can come together, and now you have a V right next to a J. Again, random. That rag bound downstream of the V1, and the second rag bound upstream of J whatever. Whatever they bound to, that's what they bring together. Completely random process. And now we cleave the DNA, and we have an opportunity now to heal that cut, and we have a V right next to a J. And so now we're going to hook them together, and we have what's called a coding joint. We have a V hooked to a J, and the other here have something called a signal joint. Actually, the only way we learned how this process worked is because we found these signal joints. We're going, what is this little circular piece of DNA? You sequence it and you find out, well, it's the variable and the joining gene segments of the light chain muscle. And that kind of got them clued into, okay, well now these two are together, so this process was discovered by identifying a signal joint that really has no physiological purpose. It will eventually get degraded by nucleases and the nucleotides used for other DNA replication. This is just another version so that you can kind of see it. Two different rag proteins that are bound to two different V and J, or in this, it could be a D and J, who knows. And bring them together, cleave it, and this is where it gets a little crazy, because you can start to add things in there, but you can see that the coding joint is one, the signal joint is one. I just wanted you to have another picture of that, and we're not going to worry about what's DNA ligase 4, what is this XRCC4, we don't care. I just want you to visualize how this is taking place. And again, this is not important for you to understand the mechanism, and why it's called P nucleotide, and why it's called N nucleotide. Just be cognizant of the fact that there is imprecise joining when a D hooks to a J, when a V hooks to a J in the light chain, and then when a V in the heavy chain hooks to a DJ that's already rearranged. So let's make sure we have this down. First is D to J in the heavy chain. Second is V hooks to a DJ in the heavy chain. And then we actually stop. We stop doing any more rearranging, we turn the rag genes off, etc. And we will go to see, did we make a heavy chain that works? And how many nucleotides code for an amino acid? 3. So if we start with, we transcribe from a promoter, and we're going all the way through the V, and we're making good amino acids, and then we've added some junctional diversity in there. What happens if we add 2 amino acids instead of 3? Frame shift. We throw it off downstream. We could have a protein downstream that's not functional. And so once we go through this process, add junctional diversity or not, then we have to test it and make sure it's going on. But I just want you to know, there's a P nucleotide, don't worry about what it means, it comes from palindrome, and there's N nucleotide that is catalyzed by the TET, that just adds nucleotides in between all of these junctions, in between D and J, in between V and D of the DJ, and in between the V and J of the light chain, which is done a little bit later. All right, so this is just showing you, and we're not even going to go through this because this gets really ugly, hair pain, lips, and all stuff, so never mind this, we're moving on. So, this is the other critical thing, and this is where I get to show you what's going on downstream. Remember I told you, just downstream of the last J segment of the heavy chain was that new constant region gene segment. So, the stuff that encodes the IG heavy chain. Just downstream of that is delta. So we look at this bottom here, we've got mu, delta, gamma 3, gamma 1, alpha 1, gamma 2, gamma 4, epsilon, and alpha 2. All of the heavy chain constant region gene segments that we need to make all of the different heavy chain molecules. But what do I have written out to the side over there? Enhancer. For most transcription to take place in our systems, you need to bring a promoter in closer proximity to an enhancer that enhances the transcription. The promoters upstream of every one of the V gene segments of the heavy chain are too far away from a promoter, from an enhancer to get any enhancing effect. There's no transcription taking place, any appreciable transcription. There is some germline that we're not going to get into. But because they're too far from an enhancer, we don't do much transcription. When we do this rearrangement, D to DJ, we loop out the DNA that was in between all of any D or J segments or any intervening DNA that was in between gets looped out. And then when we take the V upstream and do that, we loop out intervening DNA. We loop out enough DNA doing this V, D, and J rearrangement. Now the promoter is in closer proximity to an enhancer, and we can actually transcribe through. And we're actually going to start doing that. We're going to transcribe through V, DJ, and mu. But we're actually going to pick up delta where we're at. The primary transcript that would come out of this transcription would be VDJ, the intervening sequence C mu, intervening sequence C delta. So we would have in the RNA that is transcribed, we would have everything we would need to make the variable parts and the heavy chain parts of both IgM and IgD. And that will come to play a little bit later. This is showing you. The top is the DNA. It's no longer completely germline because we've done a V, D, and J rearrangement. DJ first, and then the V to D. But you can see up there that we have all of the stuff we need for C mu. We have some materials that are necessary for making a secreted version versus a membrane-bound version. And then we have the delta and the same thing, membrane-bound versus secreted versions. And it's actually, have you all heard of alternative splicing? So when messenger RNA is made in eukaryotic systems, we'll just talk about mammalian systems, there's those intervening sequences. We have introns and exons. And the primary RNA transcript still has that in there. You have to splice out the introns before you have the function, the real messenger RNA that goes to be translated into a protein. You can have alternative splicing taking place. And this is how you get some diversity in several molecules that are in mammalian systems. In this case, you can actually have an alternative splicing event where instead of splicing the V, D, and J to the mu gene segments, it's spliced to the delta. So on the left side of the panel, we're going to end up making IgM because the splicing event brought the IgM constant region gene segments in close proximity right up there against the VDJ instead of the delta when the delta is gone from that transcript. So we make IgM. And the other one, we alternatively splice down to the delta, and now we have the delta, IgD. Did we change the affinity of the molecule at all? Somebody say no. No, we did not, because we still have the same VDJ. All we did was change the heavy chain constant region gene segments that are associated, so effector function has changed. You go from doing what an IgM would do to doing what an IgG does. And in fact, in B cells, they put both IgM and IgG up on their surface, even though IgD doesn't play a role for what we care about. So, same specificity. We're just changing the constant gene segments that are associated. That's not good enough. B-cell antigen receptor doesn't have the signaling that it needs to send a signal from the antigen once it's bound. So we have to have, remember when I talked about ITAMs a while ago, the immunoreceptor tyrosine activation motifs that we saw on CD3 of the T-cell receptor. We also are going to have those with some molecules that are associated with the B-cell receptor. And again, scientists like to use Greek letters, so we've got Ig alpha and Ig beta, and that can get very confusing because we call IgA Ig alpha and don't, okay? These are different molecules. These are signaling molecules that are associated with every B-cell receptor, this immunoglobulin that goes to the surface. They are the part that actually signals, and they have the same ITAMs, or they have ITAMs associated with them that are involved in signaling to it. So this is a full B-cell receptor, but so far all we've done is made a heavy chain. We haven't made a light chain. That can't go to the surface. I'll just take my word for it. The heavy chain can't go up to the surface without some type of light chain. But we haven't started rearranging the light chain yet. So the cell does something really clever, and it makes a fake light chain, what they call a surrogate light chain, and associates with it. Now, remember, you have two sets of, because you're a diploid organism, you have two alleles of heavy chains. You have one from one parent and one from the other parent. But you can probably rearrange both of them. The RAG genes got turned on and they start doing their thing and they're trying to rearrange. Well, we don't want to have immunoglobulin, we don't want to have heavy chains of two different specificity that interact with light chains to give us a multi-specific B cell. That could be disastrous down the road, and there's other reasons for it. So we go through allelic exclusion. I hope you've heard of that before. Allelic exclusion simply means one allele is being used and the other one is being excluded. It's just not being used at all, turned off. And there are various mechanisms for doing that. But once we rearrange the VDJ of the heavy chain, we want to turn off the other one, the other allele. Well, how do we do that? We have to get some kind of signal that we have rearranged the heavy chain. You need to stop making any more of that. And so that's why I showed you the previous slide with the signaling molecules. What we're going to do is we're going to go through, what did I do? Oh, that's, okay. I took some of it out, that's what I did. What we're going to do is we're going to go through a process of sending the molecule up on the surface to make sure that we have a functional IgM. I'm going to see what I did here. I tried to cut some of the detailed stuff out. And I took all that out. So what's going to happen is and you'll hear about this in the next part of this particular thing, the B-cell maturation phase. The heavy chain is going to go up on the surface, and if it's functional, that is, there's not a frame shift mutation, and it has structural components that are good, and it can recognize anything out there, it's a functional heavy chain, then we turn off the RAG genes, and the chromatin at the other allele gets altered so that there's no, actually both alleles, so that there's no more rearrangement of the heavy chain. So the allelic exclusion gets kicked in, and we know we have a good heavy chain. And now we're going to actually go through a stage of we've gone through a lot of work to get something that works. A lot of times it doesn't work. You don't get a functional protein that can go to the surface, or it doesn't get a signal once it goes to the surface. And that B cell is going to try again, but a lot of times it's going to die. But once you get one that's functional, you want to make sure you do a lot with that. So we will proliferate. We'll go through 5 or 6 rounds of proliferation. So you do calculations 1 to 2 to 4 to 8 to to 16 rounds. So you're gonna go through about 5 or 6 rounds of proliferation, and you have clones. Now, there may have been some slight mutations in them to make them, but for the most part, you're gonna have, 30 to 60 clones, all of the same heavy chain. And then the RAG genes get turned back on, and we start going through the processing again with the light chain. And it could be kappa, it could be lambda. What ends up happening is let's just say 60 clones start rearranging the light chain loci, and you're probably gonna get 60 different things because it's a random process. I just need to reiterate as much as I can that this is going on in your bone marrow all the time. And it is a completely random antigen independent process. The B cell doesn't care what it recognizes. The RAG genes don't care what the gene segments are that they're binding to. They're binding to gene segments, they're bringing them together, and we're going to see what happens. And it's going on all the time. Yes?
So just so I have this kind of straight in my head, the heavy chain, once it's functional at the surface of the cell, a lead exclusion happens, and then proliferation of that
Yes.
specific.
Of that clone will then proliferate. And it's fine that that's up on the surface. It doesn't do anything. And eventually, everything that goes up on the surface will eventually come back down. So over time, the membrane is a dynamic thing. And so what's going to happen is those things are going to come off of the surface later. After we get a light chain that works, after we go through the VJ rearrangement of the light chain, it associates with the heavy chain. And we'll just put this up here. We have the heavy chain. We have the heavy chain, and it's functional because it's associated with these signaling molecules, Ig alpha, Ig beta, and it has a surrogate light chain. It's every one of them have the same exact thing. It's a surrogate light chain. It's just something that looks like a light chain that associates with it. So you've got this Y-shaped molecule with these signaling molecules that goes up to the surface. We get a signal to the cell. We turn off rearrangement of the heavy chain. We proliferate 5 or 6 rounds. So we've got all these clones. We have 60 clones hanging around. All have the same heavy chain. Now we start rearranging the light chain, and it's a VJ rearrangement. Once we get one of those that looks like it's working, it's productive, it'll associate with the heavy chain and try to go to the surface with Ig alpha and Ig beta. And the surrogate light chain goes away. So we get a light chain associated with a heavy chain. Actually, it's 2 heavy chains, 2 light chains, Y-shaped molecule, signaling molecules go to the surface, and now we're going to go through the process of seeing, do we have a fully functional antibody molecule? A lot of work to get to the point where we have something that may recognize an antigen or it may not. And it may actually recognize one of our own antigens, which is not a good thing. So we go through the allelic exclusion, we go through the process of making the light chain, getting it up on the surface, and we're going to talk about the maturation of the B cell a little bit later. Now, what's going on there? Actually, we might jump right into that right away. Let me see. I think we're going to just get right into what's going on. If we go through that process, we end up with a B cell that has an immunoglobulin molecule on the surface that can recognize something. And about later on, there's a few slides that goes into the order of rearrangement, what's going on, and there's a selection process. We want to make sure we shut down those B cells that recognize self. And so we have 2 types of tolerance that we talk about. There's the essential tolerance, and in the case of the B cells, that's in the bone marrow. They are going to sample self-antigens that are located in the bone marrow. If they react way too strongly, we're going to delete that B cell. We're going to send it in anergy or apoptosis. We get rid of the self-reactive B cells, but some of them get out. Not all the self-antigens are present in the bone marrow, so some of them get out, and we have additional peripheral tolerance mechanisms to take care of that. But the next piece that we were kind of going to move into, I wanted to, was, all right, Remember when I said we had alternative splicing from IgM to IgG? We also have another event where we can alternative splice between membrane bound versus secreted. So up until this point, all we've done is made a membrane bound form. And we're just gonna jump ahead and say we have a good heavy chain, we have a good light chain, and it's up on the surface, it's a membrane bound form. But we can form, after we encounter antigen, we go through some processes, and I wanna just put this up here. This is a B cell that has gone somewhere and is interacting, it's going to interact with antigen. So you have the B cell, and it has IgM and IgD on its surface. A little bit of it, just because it's alternative splicing, you're going to get both of them on its surface. Doesn't affect specificity. They're both the same specificity. So this B cell, let's just send him into the lymph node, hanging out in the lymph node, and all of a sudden, its specific antigen comes into the lymph node and they interact. That causes activation. And then what's going to happen next is that signal for activation, it's interacting with this cognate antigen, says there's something out there that you need to bind to. You need to make antibodies and bind to it. So the first thing it does is it proliferates. And right now we're just talking about one clone, and for our purposes, that's one of those 60 clones from long ago when we first developed our immunoglobulin heavy chain. So one B cell clone recognizes its antigen in the secondary lymphoid tissue, it's activated, it proliferates to some level, and then we have differentiation. And what we're going to talk about is, one of them is the plasma cell. The plasma cell is going to start forming, and it's going to have the ability to make a secreted form. And that's what you have on this slide here, because the materials just downstream of both IgM and IgD, actually let's just focus on IgM, has the ability to make the membrane-bound form or a secreted version. And it's an alternative splicing event that is involved in that. And so on the left side of your screen there, it's showing a membrane-bound form, and on the right side, it's showing the secreted form. A alternative splicing event, and now what's going to happen in plasma cells is they completely convert to making the secreted form. They may have some membrane-bound form on the surface for a while, but eventually it's going to get pulled in. They're just going to make antibodies. And I think the half-life of this is about 3 days. Plasma cells just chug out antibodies for about 3 days, and then they start to poop out. All right, what if the antigen is still present? Well, the antigen is still going to go back to these B cells that you still have here, and some more of them are going to differentiate into plasma cells to start making more antibody. So that's one differentiation event. The other differentiation event is the memory aspect. Let me see what's next here. So I'll move on from there and show this next thing is diversification after antigen encounter. So some of these cells that proliferated right here are going to differentiate into plasma cells and go make antibody. Some will go through another type of differentiation and become memory cells. But what happens every time we proliferate? One thing different has to happen, that is the DNA has to replicate. And anytime we replicate DNA, we have this potential to introduce mutations in the 6 CDRs. So what's going to happen also is this affinity maturation piece. By introducing individual mutations in those hotspots, you could change one amino acid in this place. One amino acid there, 2 amino acids, one in each place. What ends up happening is you alter the pocket where the antigen is binding just a little bit by these point mutations that you make. Some of them are going to be better, have higher affinity, and some are going to be lower. So the slide says we have As antigen levels decrease, B cells that bind low levels of antigen are selected. Higher affinity B cells are going to continue to be activated because they have a higher affinity for the antigen that they have. Those lower affinity B cells, they're going to kind of poop out. They might be able to hang around a little bit, but they're not the ones that are going to really eventually end up over here as a memory cell. The higher the affinity, the more likely that B cell is going to end up being a memory B cell, so that the next time you come in contact with an antigen, they respond very quickly. The memory B cells respond much faster. No lag phase, there's also more. So we've gone from 1 in a million B cells that recognizes antigen A to now we have, I'll just say 100 in a million that recognizes antigen A with a very, very high affinity and will respond at a very low concentration of the antigen, and so you respond much more quickly. All right, what does that look like? I'm going to put both of them up. Okay. 1 week after primary immunization, this is just looking at the 3 CDRs in the heavy chain and the light chain. This would be like a typical experiment where you go in and you look at the amino acid sequence of the heavy chain V region and the light chain V region of a mouse that's been immunized with a particular antigen. And so 1 week after primary immunization, you still have IgM. You're still making mostly IgM. You haven't done a whole lot as far as the affinity maturation, as far as the differentiation. You've made plasma cells that are IgM. The longer you go, the more chance you have of doing this isotype switching, which is coming up. So we're mostly IgM, because that's the first one we make, IgM. And we can start to see that there's a few, those red boxes are going to be point mutations in the heavy chain V region and in the light chain V region after 1 week. You go out 2 weeks, now you've had some isotype switching. It's some IgM, some of them are producing an IgG molecule now, which is actually, what do we know? It's better for what we want to clear something in an infectious agent if we have some kind of pathogen. The IgG is something we run a little bit better, it's a better antibody. And we see a lot more mutations taking place there. If you went out further, you would see even more. So those hot spots of mutation going on in those loops of the antibody molecule are changing the affinity of that particular B cell for the antigen. If the affinity is better, that B cell continues to be selected. If the affinity is lower, that B cell is no longer selected. Only the best survive. And then this is just getting back at, remember that the first thing to come out, the first isotype that's being produced is IgM. What happens also during all this process, activation, proliferation. During proliferation we have affinity maturation. We have differentiation. We have plasma cells that go make the antibody. So a lot going on here. And we also have memory being produced so that this vaccination is going to be lasting. We also have isotype switching. And we won't get into the mechanism, you're just going to have to trust me that this actually happens. But because we have all of the material downstream in the DNA, we have all of the conservation genes for all the different isotypes downstream in the DNA, we can switch to them if we get the proper signal. And so this is kind of showing you what it would look like. Upstream of every one of them, except for delta, you see a switch region. S, and then a mu is blue, S gamma 3 in yellow, etc. Those are switch regions, and what happens is, during an immune response, depending on what kind of cytokine environment the B cell is being exposed to, proteins are going to be, the cytokines will bind to the receptor, and proteins are going to be sent to the nucleus, and will cause some of these switch regions to start hooking up, and we switch to a different isotype. Yeah?
When does the isotype switching occur? At what point in that step?
It would be down here during differentiation. So it would kind of be all through here, but it's really going to be down here when it's in this differentiation phase. And it may or may not happen. It all depends on what kind of environment is there. If there's IL-4 and IL-5 present, we get IgE switching. So I'm not sure, this one is showing switching to alpha,
So it occurs before it becomes a memory cell or a
Yes.
plasma cell?
So you're gonna have some memory cells forming with the IgM, but they're not great, so they probably won't be, they won't be signaled, they won't be selected the next time you don't run into it. But if we go through affinity maturation, we go through isotype switching, so I'll put that right here, then those cells will be the ones that will have the highest affinity antibody on their surface. But suffice it to say, proteins, much like the RAG protein, but not amino acid sequence, they pull the DNA together at these switch regions and loop that out. That B cell clone now no longer can make mu, delta, gamma 3, or alpha 1. Is that right? I can't read what it says. All that DNA up there is looped out, and so alpha 1 can now be made. It's downstream. That's kind of, yeah. That's gamma 1. Love it. And so that DNA is looped out and cut, and that B cell can no longer produce any of those other types. They can switch again if it comes in contact with a different environment. You can bring some different switch regions together, and maybe you'll switch down to alpha 2 or Z down to E, to the light blue down to E. That would be an allergic reaction. So what would typically happen is, in certain cases, you would have cytokines like IL-4 and IL-5. They are the two cytokines that are involved in causing the B cell to switch to making IgE. And so this is where this would happen, is you would loop out all that other DNA, and all you have left is the IgE and whatever else is downstream of it. And you would be making IgE from that particular B cell clone. And all the plasma cells that would come out of that B cell clone would be making IgE of whatever affinity that thing had. And as we mentioned, in the case of IgE, it would immediately go and bind to an Fc receptor on a mast cell and sensitize that mast cell for allergic reactions. Okay, let's summarize. So this just showed switching to alpha, and there's the dimeric form of alpha that we saw before. So now you're making this instead of a pentameric IgM. Let's summarize this. The first thing that happens is the heavy chain. And the first thing to rearrange is one of the D genes is going to rearrange to one of the J genes. So you can see if we're in the germline situation, we have what's called somatic recombination. The RAG genes were involved in binding to the recombination signal sequences that have those spacers, 12 and 23 base pair spacers. Brought a D gene in context with a J gene segment, cut the DNA. The DNA that was in between was looped out as a signal joint and lost from that B cell forever. Then there could be some imprecise joining. We could have P-nucleotide addition, we could have N-nucleotide addition. All that does is add additional diversity if the protein is still producing a constant region genes downstream that are in frame and are functional. So typically you're talking about then nucleotides of 3 would be how it works. It's a little different in T cells, but we'll get to that. So 3, 6, 9 amino acid changes. 3, 6, 9 nucleotide changes. We had 1, 2, 3 amino acids. Once the DJ is rearranged, then one of the V gene segments is brought in close proximity, and we do the same thing again. RAG 1 and 2, we cleave the DNA, bring them together. We could have some junctional diversity added or not. It's all random process. And then we have the possibility of putting IgM up on the surface. The promoter is now close enough to the enhancer. We do a primary transcript through that. We splice out the intervening sequences. We have a messenger RNA that is translated into a protein that can do nothing. So it associates with a surrogate light chain and with the Ig alpha and Ig beta signaling molecules and goes to the surface of the B cell. Once it's up on the surface of the B cell, it does these other things where it gets a signal that says you have a functional heavy chain, so now start rearranging the light chain. And so now we go to the left side and we do a V rearranges to a J, and when we have a functional light chain, the light chains associated with the heavy chains associated with the Ig alpha and Ig beta and go to the surface, and we should have a functional B cell receptor on the surface of the B cell. But that's not always the best thing. So as far as we look at the summary here, multiple B segments of heavy and light chains, multiple J segments of heavy and light chains. These segments add diversity to the heavy chains, but not the light chains. And so all of that combined itself, recombine the different versions you could have with VD and J and V and J of the light chain, you could have over the 2 million different kinds of specificities. But you also have the junctional diversity that's added in there. So all of these things contribute to junctional diversity, or diversity of the antigen binding site. You also have various associations with different heavy chains, different light chains, you have kappa and then later on, we have this somatic hypermutation that takes place, which is called affinity maturation. Anytime the B cell replicates, anytime it proliferates, makes more of itself, the DNA is replicated, and therefore those hot spots that we talked about, CDRs, you could have a mutation added to them. That could also change the antigen binding affinity of the molecule, adds more diversity. All of this is about adding diversity. And then you could have, the last thing here is saying isotype or class switching. It's all capital underlined. Isotype or class switching, which is moving from an IgM to an IgA or an IgE or an IgG, does not contribute to antigen binding affinity. It's just that's in the effective function. All you're doing is altering the effective function of the molecule and go along. These last things we're going to go through very quickly because it just kind of goes through this again and shows you what's going on in a different way. This shows you a B cell. The first panel is in the bone marrow. It's associated with a stromal cell. It's going through the process of making an antibody molecule or an immunoglobulin molecule. It puts it out, it's up on its surface, and you're going to see, this is the first time I've used the term negative selection. So how do we get rid of self-reactive B cells? We've made an IgM, put it up on the surface, it's IgM and IgD on the surface, and it turns out it's very reactive with one of our own antigens. We don't want that getting out. Well, it can bump into those antigens in the bone marrow, and those B cells will be deleted by apoptosis. We can recycle the materials and make something else out of them. If the B cell has a functional antibody molecule, immunoglobulin molecule, but is not self-reactive, it can leave the bone marrow and go in search of its foreign antigen in the secondary lymphoid tissues. And so they migrate to the peripheral lymphoid organs. You can see if they have IgM on their surface and they have IgD. So this is what we call the naive B cell. It has never encountered its own specific antigen. It has on its surface IgM and IgD. If it had encountered its self-antigen in the bone marrow, it would actually decrease the IgM and just be IgD positive and would be allergic or would be deleted. That's the role of IgD, is just to make sure that we get rid of these self-reactive B cells. So we've got this thing, and I just bring that up because if you look at this picture, it looks like you've got two different kinds of antibody on there. You do. They have the same affinity. You have IgM and IgD through the alternative splicing. Once they come in contact with their antigen, all this stuff takes place. Activation. Proliferation is the first thing. What can happen during proliferation? You can get the somatic hypermutation that takes place, the core mutations that can affect the affinity of the immunoglobulin molecule. You can differentiate. You can have, under certain cytokines, you can start switching to a different isotype. You can have some of them that become good memory cells, and it could be some of them still with IgM, but mostly it's gonna be those that have gone on to become an IgG type. And then the plasma cells, the effector cell. These things stick around. The ones that have gone through affinity maturation and become memory cells, they're gonna stick around for life, or somewhat like life. And the plasma cells don't. They make their antibody and they die, and we don't see them anymore. But that whole memory cell generation is a whole different story. Give me about 2 more minutes. Might have to take a little bit more than that. B-cell maturation, I don't expect you to be able to reiterate this to me. This is just allowing you to see everything I've said about 5 times in one slide. Germline versus the DJ rearrangement of the heavy chain, and you can see nothing going on with the light chain. Then VDJ rearrangement of the heavy chain. Once that's rearranged, we add a surrogate light chain. We find out we have a good heavy chain. Then we start rearranging the light chain, and then we have a functional light chain. And we have IgM and IgD on this mature B cell that leaves the bone marrow and goes looking for its antigen. This is just showing you the chelating that I've mentioned several times. IgM on the surface comes in contact with the multivalent self-antigen in the middle, and it goes through apoptosis. If it comes in contact with a soluble self-antigen, it can survive, but it becomes emergent. And the term emergent just simply means it's unresponsive. It's not going to be able to get life signals forever, so it's eventually going to poop out and be taken up by some phagocytes and degraded, or by some other thing. So any type of a very strong interaction with the self-antigen is going to kill the cell, kill the B cell. A weak but a good enough signal against another self-antigen would just cause the B cell to become emergent. Here's just showing that what we've seen before. The high endothelial venule is in the blue area of the lymph node. That's the T cell rich area. So the B cell would leave the bone marrow and start going into the lymph node, and they have to travel through the T cell rich area up to the B cell rich area. And if they're not getting what they want there, then they can go out and they leave and go to the next lymph node or go to the spleen or wherever. So they start circulating through the lymph nodes. And is that right? Yeah. Further maturation and activation, these are the things that I talked about down here. They encounter their antigen in the T cell rich area, they can differentiate into plasma cells, and those first ones are going to produce lots of IgM, because that's the first thing they produce. We can also have migration of the activated B cells into these germinal centers, those B cell rich areas, and that's where the affinity maturation is going to take place due to replication and the mutation of the CDRs, isotype switching will take place, and memory B cell generation takes place. Do not ask me how memory B cell generation takes place, because I don't think anybody has a real good idea what's going on there. But we do know that these B cells that do become memory B cells are long-lived. It looks like they're long-lived, some of them 20 years, but we may actually be wrong in that. It may be that at some point there may be a follicular dendritic cell that sequesters a little bit of the antigen every now and then, gives it a little bit, and proliferates it up again, gets a few more new clones so the old ones can die. We don't really know, but it certainly happens, because the next time you're exposed to the antigen, you respond much more quicker, the antibodies are better, they last longer, and they are made at higher level. So, we're going to stop there. The next slide just kind of shows you that in a diagrammatic form, what I had written in the text.